In the last video, I mentioned a little bit about how microbes play a role, especially in things like decomposition. And one of the reasons that that's really important is because it allows for the recycling of nutrients in the ecosystem. A really important nutrient that people don't always think about, you think more about carbon than you do about nitrogen. But nitrogen is critically important to make both proteins and nucleic acids, like DNA and RNA. One of the problems is that while there's lots of nitrogen in the atmosphere, unlike carbon, there aren't many large organisms, like plants, that can pull nitrogen out of the air and turn it into a form that can be used by us, for example. With carbon, plants can take carbon, they do photosynthesis, and they can make carbon-based nutrients. So how do then we get nitrogen into our bodies so that we can make our own nucleic acids and our own proteins? Well, we rely on bacteria to do a process that's called nitrogen fixation. And what that means is they take nitrogen from the atmosphere in the form of N2 and turn it into a form of nitrogen that we can use. They can also do this with sulfur. I also talked about how microbes are involved in digestion. And so microbes in the guts of cows break down plant compounds like cellulose into carbon dioxide and methane. And those are the gases released from cattle. So again, without those microbes, you're not getting the digestion. So microbes are really important in agriculture for both plant growth and for working with animals. There have been a lot of studies done where soil that does not have the microbes that can do this nitrogen fixation process don't support good plant growth. The plants don't grow very big. They don't produce, especially we're talking about legumes, so plants like beans and soy, they don't produce enough of the product if they're not associated with these bacteria. So really important in agriculture. In the environment, we can talk about the idea of biofuels, for example, turning corn into ethanol that maybe we could use as a type of clean energy to run vehicles. We talked about the idea in the previous video about doing bioremediation or using microbes to clean up oil and other pollutants. Microbes can also be used in mining. There's compounds like copper sulfate, and you can use microbes to pull out copper that you can use for whatever purpose you need copper for. This is again showing nitrogen in the atmosphere and how you have to have these nitrogen fixing bacteria to pull nitrogen from the atmosphere and put it into plants so that it can be eaten by animals, decomposed for other bacteria and fungi. I'm not going to ask you the details of all of these processes. I know this is kind of a complex cycle. But again, the idea is that ability to fix nitrogen, to pull nitrogen gas out of the atmosphere and turn it into another form is very, very important. We use microbes in food. Sometimes we want to get rid of microbes, so we think about food preservation. Maybe we heat our food to kill any microbes. We keep our food cold to prevent the growth of microbes. Lots of our foods, like spices and fruits, are irradiated to get rid of microbes. They can also be treated with chemicals as preservatives to help keep microbes from growing and keep our food from going bad before we get a chance to eat it. We can use microbes to ferment foods like cheese, beer, and other examples. And we can use microbes as food additives. MSG, citric acid, yeast are all, well, yeast are a microbe, but the monosodium glutamate and citric acid are produced by microbes and used as food additives as well. In biotechnology, we have the use of microbes to make genetically modified organisms, or GMOs. GMOs are not a bad thing necessarily, but they can, especially in plants, allow plants to be resistant to herbicides or weed killers, so that a farmer could spray his farm with a weed killer, but his plants that he's growing won't die. You can also have plants that are resistant to being eaten by insects. They make a toxin that is bad for insects but doesn't hurt mammals or people. So genetically modified organisms have allowed better crop yield, better crop production. They can add nutrients to crops like vitamin A in areas where people don't have access to as much nutrition. We can use microbes for the production of pharmaceuticals like insulin and other human proteins. And I mentioned CRISPR before. Scientists are using microbes to look at gene therapy or treating people's genetic conditions. Microbes, I think we've been living through this, right? We're living through a pandemic. Microbes can be new diseases. And we can use what we know about diseases we've seen before to help us understand new diseases that have emerged. And we can use microbes to develop new treatments or cures, even preventatives like vaccines. So microbes play an important role, not only as disease-causing organisms, but also in disease therapies. Microbes are found pretty much everywhere. I'm trying to think of a place where there aren't microbes, and I can't. Anywhere there's water, and sometimes even when there's not, there are microbes. Scientists drilling in things like ice cores, like Antarctic lake ice, they find microbes at below freezing temperatures. Scientists looking at hot springs like those in Yellowstone National Park find microbes living in like literally boiling water. They find microbes at the deep sea vents where there's no sunlight. There aren't very many things for them to decompose, but these microbes live on things like sulfur and methane instead. And they can survive in these undersea vents that have really, really high pressure and really high temperature. Again, like hundreds of degrees. They can survive. So microbes can live pretty much anywhere in any kind of environment and tolerate lots of different environmental stressors. Now, not every microbe, of course, can live in every environment. Each microbe is going to be evolved for its particular environment. So you won't often see microbes that live on the seafloor on a plant, right? They're only going to be on the seafloor. They're not going to be on plants that are on the surface. If they can live in Antarctic lake ice, they probably also cannot live in an acidic hot spring. So microbes will evolve to live in the environment where they are. And that's great because now they can live in this environment, but that is the trade-off that they can't live in other environments. Over time, how scientists have classified organisms has changed. In the 1700s, when Carol Linnaeus was designing what we now use as the taxonomic system, he basically classified everything as either an animal or a plant. Basically, if it didn't really move and it was green, it was a plant. If it could move and it wasn't a plant, it was an animal. That was pretty much the classification. As we moved into the 1800s, we had animals, which were animals. Both fungi and plants were plants. And then anything that was small, like a single cell, was called a protist. Okay. In the 60s, we still had our animals, but we learned to separate the plants and the fungi. We still had this group of single-celled organisms. These are a little bit different from bacteria because they have a nucleus, and we'll talk about that a little bit later. And then we had a group called the Monera, and these were all of our organisms that did not have a nucleus. But as we started doing sequencing and looking at the DNA of these organisms, I say we like I was involved in this, but scientists created a classification system that has one level higher than kingdom. And the highest level of classification you can have is a domain. There are 3 domains. There are the eukaryotes, which have all of their subclassifications. And then there are the bacteria and the archaea. And both of the bacteria and archaea have no nucleus, and the eukarya domain has a nucleus. And like I said, the domains of life are based on DNA sequencing. So the reason that we couldn't classify them this way before was because scientists didn't know how to do DNA sequencing until the 1990s and continuing through today. So there's a particular gene that they look at that every organism has. And so by looking at that gene and looking at the sequence and comparing them, scientists have been able to tell which organisms are more related to one another. So the sequence will be, they call it alignment. So if you're looking at 3 different organisms, you sequence that little gene and you compare the sequence. And then you can generate what's called a phylogenetic tree, which shows the relationship. So organism 1 is more closely related to 3 because they're closer together. Organism 2 and 3 are very far apart because they're far apart on the tree, so they're not as closely related. And again, this is kind of what this looks like. And what's really kind of cool to see is sometimes people have called it a tree of life, where you start at the root and then you branch off into the different groups. But to me, it's almost like a bush, and it's kind of messy because you have these exchanges between bacteria and eukaryotes and archaea and bacteria that have allowed all of these different organisms to have evolved the way they did. And so again, we have this idea of the tree of life. And I just kind of want to highlight, these are supposed to be your bacteria, which are more closely related to archaea. Both of these are a little bit more distantly related from the eukaryote. Remember, these have nuclei, and we're over here, Homo sapiens. When we're looking at the DNA, especially of more complex organisms like us, generally each human, in terms of DNA similarity, each human is 99.5% similar to every other human. That 0.5% difference is what makes each human unique in terms of the way they look. But there could be parts of our DNA that don't even affect the way we look that are also different. But that's the estimate of how closely related 2 random humans might be. When we compare humans to some of our closest primate relatives, humans and chimpanzees are between 96 and 98% similar, 90% similar to cats, 82% to dogs, 80% to cows, all the way down to 60% similar to chickens and fruit flies. And you don't think that you're about 60% the same as a fruit fly, but you are. You're 60% the same as a fruit fly. With bacteria, it's a little bit different. For animals, we generally say that animals belong to their own species if they can't reproduce with each other and make an organism that is fertile. And what I mean is, for example, if a dog and a cat tried to reproduce, there would be no offspring. So they're different species. But what about a horse and a donkey? A horse and a donkey can reproduce, and you get a mule. That's the animal that results from a horse and a donkey. So why are horse and donkey different species? They produced a viable offspring, a mule. Mules are sterile. They cannot reproduce. So it's a dead end. So horses and donkeys are different species. With bacteria just make a copy of themselves. They don't sexually reproduce. They don't seek out a mate. So how do we say that bacteria are different species? Well, we do look at the DNA. And depending on who you ask, it's anywhere from 95 to 97% similarity. So if two bacterial organisms are between 95 and 97% similar, we'll say they're the same species. If they're less similar, then we would say that maybe they're different species.